mostly deterministic. We have very little infection. And then in between, we have what I like to call fuzzy boundaries between deterministic regions, where for the same parameter set and multiple simulation replicas, we get lots of stochasticity, lots of variability between simulation replicas, and sometimes major differences in simulation outcomes. Now, we went more through this last time, and of course, as I've seen dropped into the chat by Jim Sluka, thank you for that, Jim. We went into a bit more detail about some of the causes of the differences in simulation replicas for the same parameter set. And what was discussed and is most interesting is that the two main causes for differences between simulation replicas is the stochasticity of various cell-based events in the model, as well as the spatial distribution of infection. So we see from this example here on the right, these are 3 different simulation replicas of the same parameter set. And what we found was that for positive outcomes where infection was slowed, as you see here, the end of simulation, there's still at least a notable region of uninfected cells. We found that the reason for a positive outcome, as opposed to, say, an intermediate outcome, was the stochasticity at very early infection. In this case, it just so happened that the first infected cell in our simulation died. A neighbor cell was infected later, but at this time, the drug therapy simulations that we were using, this drug therapy was able to stop infection due to that delay, which was due to stochasticity. And then comparing these 2 simulation replicas in the middle and to the right between an intermediate and negative outcome, the negative outcome being when mostly all cells died at the end of simulation, we found that the difference here between these 2 was not a difference in early cell type numbers, but rather the first difference that we found in simulation was in the total extracellular virus in the simulation. And so from that we say that if the same number of cells produce a different total extracellular virus, then most likely this is due to differences in spatial distributions of infection. And then lastly, we concluded our sweep of the results that we've done so far by doing an overview of a simulation of drug therapy, as just mentioned. And these simulations would simulate something like a drug intervention that reduces the rate of genomic replication inside of infected cells. So this is the model system of viral replication, and our model describes replication as occurring, going through various stages from internalization, replication, all the way to being released back into the extracellular space. And most notably, there's an exponential term in the replicating stage. This is where genomic replication occurs and more virus is produced. And then our simulations of drug therapy, as you can see here on the right, this is a schematic of a representative example of a simulation. We said that at some point after initial infection in simulation time, we would take this coefficient Rmax, which we often call the replicating rate. At some time in simulation, we would say that a drug has suddenly been applied, and the molecular effect is to reduce this coefficient Rmax. So here is the baseline parameter value. And for this particular example, at 12,000 minutes into simulation, there is this drop in Rmax, and that is due to the drug therapy of this particular simulation. And just very briefly, we found that early treatment is better, as no surprise. You can see this demonstrated for this parameter sweep. On the left, these are early introductions of the drug therapy in simulation time. Those particular moments are shown in dashed lines. They're also annotated across the top. So these particular drug therapy sweeps went from introduction of the therapy at initial simulation all the way to about 14,000 minutes. So no surprise, moving from left to right, early therapy is better, whereas late intervention leads to negative outcomes. This is counting the number of uninfected cells this time. You can see that there's very little change in those numbers for early therapies, whereas for late therapies, we see that the number of uninfected cells goes to 0, meaning that everything, in the very least, by the end of simulation, has been infected. And then moving from bottom to top, the bottom row is the least potent therapy, meaning that the replication rate was reduced the least in these simulations. So this is a weaker therapy. And then moving up to the top, these are the strongest therapies. In this case, an absolute reduction in the replicating rate by multiplying the coefficient by 0. So on this top row, for example, wherever the dashed line is shown at that moment in simulation time, for this row, the replicating rate was set to 0. Now, you'll notice again that we have these sort of deterministic regions. So what we call positive outcomes are annotated here in green, and then negative outcomes, where most things die, or at least are infected by the end of simulation. These are annotated in red. And we have these sort of deterministic regions again, this time in the top left and the bottom right. And then again, these sort of fuzzy boundaries. Now, in the case of the previous slide with the 3 different simulation replicas, those were for this particular parameter set shown here. This was a drug therapy, a time of application of 10,000 minutes, and an absolute decrease in the replicating rate. And you can see that as you trace down, say, this column, we have lots of stochasticity, and even to very clear bifurcations in some of these different parameter sets. And then for the clinicians who might be joining us today in actual medical applications, an interesting thing that we found in these simulations was that even for, say, weaker therapies with lower potency, that early therapy was still effective. You'll see this for example, in the next to bottom row. This is a 25% reduction in the replicating rate, very much on the weaker end of the parameter sweep that we did. And for later therapies, you see that there's a deterministic negative outcome for very late therapy. But as we move to the left, even to, say, initial simulation time or very close thereafter, that we still get some positive outcomes even for weak therapies. Now, with that very quick flyby, let's start in CompuCell. So we have our nanoHUB tool. For those of you who weren't with us in the session last week, I'm going to put some links into the chat for your convenience, the links that are shown here. Now, we are providing the simulation files for those of you who would like to use local installations. For those of you who haven't installed a local copy of CompuCell yet, I'm dropping a link into the chat now for your convenience. So you can copy that and paste it into your browser of choice and select that website the installation as appropriate to your operating system. Now, as a full disclaimer, we have had some installation issues recently with Mac, so if you're on OS X, please be careful and be mindful that you may run into installation issues. In general, though, the quickest path to getting up to speed and into CompuCell development for our intents and purposes today is through our nanoHUB tool. So the second link to the nanoHUB tool has also been dropped into the chat. Again, you can just follow that link, and that should take you right to the nanoHUB tool. I'll switch over to my browser. I have my nanoHUB tool launched. I'll be using my local copy of CompuCell, but occasionally jump over to the nanoHUB tool as well. Now, when you launch your nanoHUB tool, if you haven't registered, there's a very quick registration process, and then you'll be able to launch the nanoHUB tool directly. When you launch it, it will load the simulation files for our work today. For the, well, these are the simulation files that were used in the results that we just reviewed. The workspace will be a little messy, and we'll clean that up. But for those of you who'd like to use nanoHUB, if you can follow that link and get through the process and launch the tool and get to about here, that would be great. Now, for those of you who would like to use local installation, again, if you haven't installed CompuCell yet, you can use the link that was dropped into the chat. And for those of you who prefer GitHub, we do all development for this project in a dedicated repository for collaborative development of COVID infection modeling. Everything that we do is open source, always has been, always will be, and you can download the simulation files at the link that I just dropped into the chat. That's this one here. If you download from the GitHub repository, you're not familiar with using GitHub, you go to the site and find a clone or download button in green to the right. When you click that, you'll be given this little button here. It's a blue one that says download zip. You can download that. Once it's downloaded, you'll be able to find the simulation files a little buried in all of the directories, but your final root directory for the simulation files is called model. For those of you who may be more GitHub allergic, we've also made available the simulation files on Google Drive. Now, the big messy link that's on here has now been dropped into the chat, so you can follow that there. Everything in that directory can be downloaded. That drive also contains the slides that we're using here today, as well as the slides that we used last week. So you can download those. We tried to put those together so that they're as independent of us as possible for your reference. So you can go back through last week's workshop and today's and use those for notes and references on what we've gone through, as well as what we'll go through today. For the demos that we'll be doing today, this Google Drive also contains a working copy of the final product of every basic model that we'll develop today. So for those of you who also downloaded from the GitHub repository, it may also behoove you to go to the Google Drive shared folder and at least download the folder called Demos. In the folder Demos, you should find 4 different sets of simulation files. They're all named according to the projects that we'll do today, and those are developed in the same order as what we'll do today. So for those of you who would like to go back through this later, for those of you who may get lost today and just would rather follow along and watch the magic happen, you can also download the simulation files in that Google Drive in their final state, and then you can use those for your intents and purposes. We'll give everyone just another second for any last minute downloads. Then we can get into CC3D based model specification. Again, as James just entered into the chat, if you do need any help with installation issues or finding the model files, issues with the nanoHUB, please enter that into the chat. Josh and Giuliano may they're experts at this work. They're very good at debugging, extremely helpful. And James will coordinate with you to get you connected with them to get you up to speed so you can go along with us today. All right, so let's continue with a very brief overview of how CC3D is used at a very high level. So models are specified in CompuCell3D primarily in two languages. First, using what we call CC3D ML, and this is XML model specification to do the prescription of basic details for a simulation, like the size of a lattice, initial configurations like what cells are arranged where, what plugins to use that we distribute with CompuCell, what cell types you would like to define, so on and so forth. And then secondly, we do lots of more detailed model specification for CC3D based models using Python. Now, this is for doing model events, model specification, simulation analysis at a very fine level, at the level of individual simulation steps. Doing analysis at individual, individual sites in a lattice, describing interactions between individual cells at specific moments in simulation. So the XML is more higher level simulation specification, whereas the CC3D Python is very useful for intuitive model specification of very fine level interactions. And then as we went through last time, CC3D simulation and development is primarily performed using two GUIs, the first being Player, which we'll start with today. And Player is what loads and runs a simulation. Player is used for doing real-time graphics rendering, as well as specifying some basic simulation data output, frequency, so on and so forth. And then we also provide what we call TWEDIT++, or I just call it TWEDIT. And TWEDIT is an editor for doing model specification with actually writing the scripts. So TWEDIT is designed to support CC3D based modeling with lots of built-in support features, some of which we'll use today for rapid development, say by providing templates for built-in code snippets to do specific actions like doing loops over the neighborhood of cells, adding graphics windows, so on and so forth. So we'll be working with both of these today, much more in TWEDIT this time than we did last time. And TWEDIT's what we'll use today for doing our model specification. Of course, all of this is done with XML and Python scripts. So for those of you who may prefer something a little more advanced like PyCharm, of course, all of this is supported in those environments as well. But we'll use TWEDIT for today. So I have some quick links here. We spend quite a lot of time trying to write helpful manuals to facilitate model development and model specification in CC3D. We do this primarily by keeping online manuals with read the docs. So we have a quick reference manual that's just a very sort of high-level flyby of some basic commands that we find are most useful for just about any sort of model development. We also provide a quite lengthy online manual for Python scripting. So this is a go-to resource for descriptions of what commands are available, what classes are available for CC3D Python development. And then our CompuCell3D reference manual is a description of available plugins and other available features using CC3D ML, so the XML specification. And this begins all the way from basic model specification of lattice sizes and number of simulation steps, all the way through the details of individual plugins. Jim has added links into the chat for these. Thank you, Jim, for doing that. I forgot to add them to my list. It's much appreciated. So you can quickly just grab those links in the chat and maybe save those for later, for when you come back around to, say, building your own models. So this is a set of code from an example model specification using CC3D ML. And as it turns out, these are the first few lines of the CC3D ML script for the viral infection model framework. Now, CC3D is backed by C++ libraries that do a lot of the real grunt work of the computational work in the simulation engine. And when doing model specification in CC3D ML, a lot of what's actually going on is passing information to the C++. So the actual simulation domain is managed in C++ and so on and so forth. C++ tends to not be nearly as facilitating for doing rapid model development and model specification as opposed to a language like Python. So we've wrapped all of our C++ in Python for doing more detailed model specification, and then you can interact with C++ using the markup language. We won't work with CC3D ML today, but this is part of the basic procedures of setting up any simulation. So as you go off after today to go start building your own simulations, this is actually where you'll start. Where we'll be today is working with Python steppables. So Python steppables in CC3D Python are the basic Python class that we use to do model specification. And an example is shown here on the right. This is a basic steppable. This is from the CC3D implementation for the viral infection model framework. And what you'll notice here is that there are functions defined on the steppables: init, start, and step. Now, this first function init is just used to initialize the steppable, and we won't really work with that much today. But what we will work with are these two functions, start and step. Now, any steppable that's defined for a CC3D based model will have these functions called by the simulator, and the simulator will simply execute whatever you've defined in these two functions, start and step. The same is true for init, but that's a property of Python. What's done here is that the simulator, during initialization of any particular simulation, will execute whatever you define in this function start. And you can see here there's some specification of just some basic initializations for doing chemotaxis in simulation. This is executed once during initialization and then never again. Whereas this function step, what this means is that it's executed at a simulation step. This is why we call them steppables, because this function at every simulation step is called, and whatever you define in it is executed at every simulation step. And this is primarily where we'll work today. We'll define a couple of steppables to build a couple of basic models. We'll do a tiny bit of initialization work, but primarily what we're going to do is we're going to define interactions at the level of cells and particular sites in our lattices, and we'll define events and interactions per simulation step. So this is a bit of a review for those of you who were with us last time. The simulation files for the viral infection simulation framework, these are all named with this convention of what we call viral infection VTM, which is virtual tissue model. And the entire framework consists of a set of scripts to try to build a modular and extensible architecture so that as we and collaborators and others continue to develop this model with all of the complexity that's involved with viral infection, that they can simply just add scripts without doing any modification to the existing framework, and can just plug in new models, new interactions, new events, so on and so forth. The basic framework that we've set up consists primarily of these. We played a bit with the input script for actual model parameters last time, which is the script viral infection VTM model inputs.py. This is a Python script. Where we'll be today, since today we get to do coding, is in the script that defines the main steppables of the current framework, which is in the script viral infection VTM steppables.py. Now, there are quite a few steppables, and I promise not to just go through and read off what all of these do, but what you'll notice here is that there are lots of modules that define lots of interactions. And again, for those of you who may have missed the discussion last week, where we went through these a bit more, there are recordings of those sessions available on YouTube. The links are in the chat somewhere. I'm sure that they'll be dropped in again soon. So for a description of what modules are actually represented here with these names, you can refer to those videos. Now the steppables, you'll notice though, correspond to the individual modules. There are some basic analysis steppables here, one called sim data steppable. This does actual simulation analysis. But the other steppables, viral internalization steppable, this corresponds to the viral internalization module. And we will do model specification in the same way today. And this is the method of model specification that we encourage in developing this simulation framework. Going forward, this is the methodology that we follow and that we encourage to try to maintain as much modularity and robustness as possible. So today we're going to add to this list with a couple of steppables. And that brings us to our first example. Now, before we get to this, let's do a quick overview and make sure that everyone can get the simulations loaded both in Player and in Twedit. Now, I'll start with the local installation for those of you who installed CC3D on your local machine. Now, we're going to start in Player. You should be able to open your Start menu and just type CompuCell3D, and you should get something like a shortcut pop-up. And that should launch CompuCell3D Player, where at launch you should just see a blank screen with a couple of open workspace areas. Now, to load a model specification project, we want to load a CC3D file first into Player for execution. And you can do this, the nanoHUB users don't have to do this currently since it was previously loaded, but we'll go through that for future use in other projects. In both cases, you can go to the menu bar and go to File. And then the first menu item, Open Simulation File. Now, when you click this, you'll want to navigate to wherever your simulation files are located. For those of you who are using the local installation, whether you download it from GitHub or from the Google Drive shared directory, you'll want to navigate to wherever this CC3D file is located, called viralinfectionbtm.cc3d. Now, when you've located that, you can just click it. This is the file that specifies all of the project files for an entire project model specification. You can select that and click Open. Now, if you were successful, then you'll notice on the top should see the name of the simulation script, viralinfectionbtm.cc3d. Now on nanoHUB, when you launch the tool, the simulation probably started automatically. If it happens to still be running, you can go ahead and stop that simulation by clicking this Stop button. It'll be the fourth button along just under the menu bar. Once the simulation is stopped, then you can load a new simulation file. For nanoHUB, you can access the same prompt for opening a simulation file in the menu bar, File, and then Open Simulation File. Now, when you select that menu item on the left, you should see a directory with your username. When you select that, you should see a directory available called CompuCell3D_demos. You can see on my screen, I have this selected. You select the directory with your username on the left, and then the available directory CompuCell3D_demos, and can double click that. Should then find a directory called coronavirus. You can then double click that. And this is where you should find viralinfectionbtm.cc3d. This is the simulation file that's automatically loaded when you launch the nanoHUB tool. This is also the same as the simulation file that's available for local installations. So you can click Open. And for nanoHUB, you should also see the name of the simulation file in the top, viralinfectionbtm.cc3d. And we're ready to go. Now, Player has a couple of simulation controls to be able to pause, start, stop, so on and so forth, a simulation that's been loaded. So just under the menu bar in the top left, you'll see a set of 4 buttons. When you hover your mouse over them, from left to right, you should see that they're called Run, Step, Pause, and Stop. Now, Run, as the name implies, runs the simulation. Steps through the simulation, one simulation step at a time, all the way to the end, as specified in the CC3D ML. The next button, Step, will simulate one step. And let's click that. When you click the second button, Step, you should see some windows pop up. Depending on whether or not you've run this particular simulation file yet, your screen may look different from mine, and that's fine. You can set yours up to look something similarly to mine, or whatever you may be interested in. But what you'll notice here is that after having clicked Step once, the first simulation step was executed, along with all of the initialization routines before it. And then it pauses. Now when you click Run, whatever screens are open in Player currently, you should see something start happening. You should see things start moving. Now you also should see the third button in the top left become blue, and that's the pause button. So you can pause the simulation, not stop it and terminate it, but just pause it by clicking pause. Now this simulation is still loaded and is essentially frozen. And we can do, say, particular things with the steering panel. If it's been defined, we can change parameters in real time or so on and so forth. However, you've set up your specific simulation files. We are not playing with the steering panel in this session. For more on that, for anyone who missed the last session, you can watch the videos to see some demos on the steering panel. We're going to do everything programmatically today. But after having pressed pause, if you just press Run, the simulation will continue. Fourth button is Stop, and this stops a simulation. Now, to set up something like, say, what looks like my organization of Player, we'll do this in nanoHUB. My nanoHUB tool, when I launched it, was pretty messy. I'm going to hit Step on my nanoHUB tool and see how everything looks, and then do a little organization and pick some results that are particularly interesting to me. Again, you're free to set up whichever results you prefer. Now, I have a couple of big screens and a couple of small screens and a steering panel. I'm going to close my model editor, which is on the left, with just this X button to make a little more space. You can also change the actual screen size of the nanoHUB tool in the bottom right. Now, I'm going to select any one of these graphics windows, and in Player, in the menu bar, go to Window, and Tile. So the menu item, Window, Tile. Now whatever screens you've created will then be organized into something like a grid of tiles, just to be able to at least catch a glimpse of everything that you've set up and you're rendering. The nanoHUB tool comes configured with a particular set of graphics windows. The local installations of Player will remember the configuration that you've set up in previous sessions if you have. And then you can close any of these if you please. Say, I'm not interested in the virus field, so I'm going to close my virus field. Now to create a new graphics window, see this button under the menu bar. When you hover over it, it should read New Graphics Window. Lexi, I see your question. Running the local model and don't have the panel showing agent counts. We will get to this very quickly, or very, very soon, I promise. Now to create a graphics window showing spatial distributions, again, you can click this button, New Graphics Window. Should be somewhere near the window in the menu bar. When you create a new graphics window, you should get a new graphics window. Now in that new graphics window, and any graphics window showing spatial results, you'll find this drop-down menu. In my case, it says Cell Field. Probably says that for any new ones that you create as well. When we select that, you'll see that there are some results that you can select from. Now, I'm going to, in my window, I'm going to plot the virus field. Now, this again is a quasi 2D simulation. So we have two sheets where different cell types are simulated. In the first coordinate of the sheet is where the epithelial cells are. And we say that this is the coordinate z equals 0, in which case the plane that we're looking at is the xy plane. And then on top of our epithelial sheet at coordinate z equals to 1 is where the immune cells are placed. And in this case, for this new graphics window, this was created looking at the xy plane, which is the default. And in this particular case, this is showing the xy plane at z equals to 1. And you can tell that here. You notice that each of these three windows on my screen have equal 0 or a 1. And then you'll notice on my screen that for my two graphics windows here and here, each of these is plotting the cell field. But in my top left, I have z equals to 0, and I see a grid of cells. And this is my epithelial sheet. And in my bottom right, at z equals to 1 in the cell field, I have nothing. Because our simulations are initialized with no immune cells. The immune cells are recruited by signaling from infected cells. So what I would like to do is I would like to observe the extracellular virus field at the level of the epithelial cells, in which case I will select from the drop-down menu the virus field, and then I'm going to change my z-coordinate by clicking the down button. You can see this on my screen. I'm hovering over it currently. And when I click that, I also now see a grid, but for my virus field. The reason I see a grid is because in these plots of diffusion solutions, the borders of the cells are also shown to help orient, gain a sort of sense of connection between where the cells are and the corresponding diffusive species that they are being simulated in. Now again, we're doing quasi two-dimensional simulations, so everything that we'll do is looking at the XY plane, but this leftmost drop-down also provides rendering for looking at other planes as well as general three-dimensional. We won't use these features today, but if you are, say, building three-dimensional simulations, these are a lot of very useful features to be able to look at cross-sections in simulation in real time as they progress, as well as surveying basic morphologies. Now my new graphics window is a little messy. I like an organized screen, so I'm going to tile again. And again, to do that, as we'll do often when restarting simulations, we'll go to in the menu bar, we'll go to Window, and Tile. Now, one difference that I see our local CompuCell users have noticed is that there may be some differences between what screens actually popped up and which ones didn't for, say, plotting population data. Or in the top right, the screen called Immune Response Model. This screen is actually plotting the state variable that we use for our compartmental model that governs whether or not immune cells are introduced into the simulation or removed. You'll notice as we mess with this more and more that as this state variable S gets very positive, that more immune cells will be added. And then when the state variable S gets very negative, that the immune cells will start being removed. So you can sort of watch those interactions between those two screens. And then population data, this will plot the number of the different epithelial cell types and immune cell types in simulation time. For those of you who didn't find that screen popping up in your simulation when you started it, we can do a very quick first edit as we start getting into the code. So to make that screen pop up, if it didn't for you, we need to go to the code. And let's do that. Let's open Twedit. So you can quickly launch Twedit from Player. This is true both for local installations and nanoHUB. By using this little button under the menu bar, you can see on my screen I have my mouse hovering over it. You'll notice I get a little bit of information that says Start Twedit++. So when you click this, whether in nanoHUB or locally, Twedit should launch. And it should launch with an empty script to the right. This is where we'll do all of our coding. And then there's a little project pane to the left where we'll manage our simulation files. We won't get into this very much today, but for building more advanced simulations and managing lots of scripts, you'll do all of that over here to the left. Now we can load an entire model specification project in the same way, or at least a similar way, I should say, as we did in Player. So in Twedit, you'll notice that the menu bar is a little different from Player. Again, Twedit is for development, and actually developing the code for model specification. Now to open a project, you can go to the third menu item, CC3D Project. And when you click that, you should find the second menu item, Open CC3D Project. So again, CC3D project in the menu, and then open CC3D project. Now, again, you'll navigate to wherever you saved the simulation scripts, whether having downloaded from GitHub or from Google Drive. The nanoHUB users will go through the process in nanoHUB momentarily. The local users, you'll find your location where you saved the simulation files locally and click viral infection vtm.cc3d. Select that and click open. Now when you click open, you should notice to the left, you should see a clickable entry stating viral infection vtm.cc3d. Now let's double click that. Again, for nanoHUB users, I'll switch over to that medium momentarily. It's quite similar to what we're doing now, for those of you who haven't gotten through it yet. Select viral infection vtm.cc3d, double click it, and you should see lots of simulation files open. And these are the same simulation files that we briefly reviewed before we opened the player. Viral infection vtm steppables.py, viral infection vtm model inputs.py, so on and so forth. Now, I'm going to switch over to nanoHUB and do this again. So in nanoHUB, the same button is available to start TWEDIT from player. So in player, we select this button, do start TWEDIT. And we see TWEDIT launch. Again, empty. Same goes in nanoHUB in the menu bar. We should see CC3D project. We can select that. Go to open CC3D project. Select that. And this should look familiar from when we opened in player. Now again on the left, you should see a folder with your username. This should make available the directory CompuCell3D demos. You can double click that to open it. Again, should make available the directory coronavirus. Double click that. And this is where you should find viral infection vtm.cc3d. So we can select that, and on the bottom right, click open. Now, if you were successful, then you should also see on the left a clickable item, viral infection vtm.cc3d. And again, you can double click that. And this should open all of the simulation scripts. Again, you can expand the size of your nanoHUB tool in your browser in the bottom right, just by clicking and dragging if you'd like more space. You can also quickly jump to and from different simulation scripts in TWEDIT just by double clicking the individual files in the left that are a part of the viral infection VTM CC3D project. double click, double click, double click. Now, before we proceed, if anyone hasn't made it to this point, would you please enter that into the chat? I want to make sure that we don't lose anyone here before we even get to the code. So please let us know, and we will go back through that process again very quickly. Otherwise, we will proceed, starting with getting our population data plotting. So give everyone a minute. Okay, I see no stop signs in the chat, so I say we proceed. I'm going to jump back over to my local installation. Now, I saw from the feedback that some people weren't getting some of these plots, population data. Now, these are optional features. That can be enabled, disabled, and configured at a very basic level in the steppable script, viral infection VTM steppables. These different graphics windows can be built in the simulation scripts to do, say, your own rendering for other numerical analysis that might interest you. But for now, let's just make sure that they turn on in the first place. Now, if we go to viral infection VTM model inputs.py, viral infection VTM model inputs. You should find a script that looks exactly like mine, except perhaps line 22, which is highlighted on my screen. Now, this first block of code in viral infection VTM model inputs.py, this is basic configuration for plotting and writing data to file that we've configured, as has been interesting to us as we've built this and used this for simulating drug therapy and doing parameter sweeps. The parameter sweep figures that we reviewed earlier today, as well as last week, that data was generated by using these various preferences. Now, what these specify is the frequency at which to plot or write to file a particular set of data. In this case, at line 22, this is plotting population data at a particular frequency. Or, as you'll notice at the end of the comment, you can disable it by setting this value equal to 0. So you'll notice that there are lots of entries from lines 13 to 40, most of which are set to 0. All of these different entries correspond to different sets of data. Most of them are disabled because plotting dozens and dozens of screens can get very stressful and not helpful at all. When we actually go to do batch execution, we usually use the second options, which you'll notice start with the prefix write. In this case, at line 24, this is the second feature for population data in simulation, which is writing the data to file. Now you'll notice that this is set to 0 currently on my screen, which again, you'll notice at the end of the comment, is to disable writing to file. Now, to address the discrepancy between what maybe some of you are seeing compared to what I'm seeing on my screen, at line 22, if a screen didn't pop up in Player showing data plotting, my guess would be, if not my hope, is that plot pop data freak at line 22, this variable was set to 0. In my case, it was set to 10, and when I go back to Player, see that their data plot points every 10 simulation steps. And the same is true for my plotting of the immune response model, because as it so happens, when I go back to my model input script, and I go down to line 30, see a variable for plotting the immune response data. Something like plot IR data freak. And in my case, that was also set to 10. So you can enable plotting of the immune recruitment model, state variable, as well as the data plotting at lines 30 and 22 respectively. If those don't interest you, you can just set those to 0. Those won't be critical for our work today. If they do, you can set them to a frequency of interest. They're not particularly computationally expensive, so you can set them to 1 and make really fine looking ones. So I set mine to 1 at line 22 in viral infection VTM model inputs. Now in general, in TWEDIT, to make a change and have that change be reflected in simulation, in TWEDIT, we have to first save. And you can do that with hotkeys. In my case on Windows, just Control S. It's also available in the menu bar under CC3D Project. You click Save CC3D Project. You'll notice if anything has unsaved changes in TWEDIT along the menu bar, these little discs will be red if there's an unsaved change. In my case, I just hit the space bar in viral infection VTM model inputs, and now TWEDIT is notifying me that there's an unsaved change that currently would not be reflected in Player. And now I've hit save and everything is blue. That means that when I relaunch a simulation, any changes that I've made in any of these simulation scripts will then be reflected in Player. So let's do that. For anyone who would like to have data plotting and didn't, can set the variable at line 22 equal to a value of your choice, anywhere from 1 to 10 is what I would recommend. Do that, save the script, and then let's go back to Player. Now back in Player, if you have a simulation running, then the changes that you may have made have not been reflected in the simulation. You can hit Stop in the top left. And then let's restart a new simulation just by hitting Run. And this is the first button along the top left. I changed my data plotting frequency to 1, and I would like to see that reflected in a new simulation. So I'm going to do that. So I've hit Run. And now in my case, I'm now getting data plotting at the new frequency that I specified, which was every simulation step. Okay, so that's our first edit. Now, let's migrate over to the script that we'll be doing our work in today. And it is not model inputs, it is the steppables. And that script, again, is called viral infection VTM steppables.py. So we're going to go back to Twedit and locate viral infection VTM steppables.py along the top. You can also locate it to the left in Twedit and double click it. That'll also open the correct file. And this steppable script is where the magic happens and where we're going to do our work. You'll notice as you scroll down, just kind of glancing through it to get a bit of familiarity with how this structure looks. You'll notice from the list of steppables that we went through very briefly earlier, there's the viral replication steppable. There's at line 157 the viral internalization steppable, and so on and so forth. This goes all the way down to about 1,000 lines of code, which is not bad for as many modules as is specified. Now let's go back up to the top and then let's review what we're going to build first. We're not going to start by building a steppable, but instead by first modifying an existing steppable. And we're going to do that to build a drug therapy simulation. So this should look familiar. This is the same drug therapy simulation that we reviewed earlier and talked more at length about last week. And the basic premise is that in our simulations of infection, that at some moment in time, some drug therapy causes a global effect over the entire simulation that reduces the replication of virus inside cells. Now, since both dosage and timing are important factors in drug therapy, when we build this, what we would like to define is model inputs for this simulation that define when a dosage is applied, in this schematic at 12,000 minutes, and also how much of a dosage, how strong. In our case, we'll define how much this coefficient Rmax, which controls the amplitude of exponential growth inside cells, we will define how much that coefficient is reduced whenever our therapy is applied in a simulation. And so let's do it. Now, we will go down to line 133 in viral infection VTM steppables.py. We're going to migrate down to line 133. Now here, what we would like to do is define a couple of lines of code in the viral replication steppable that introduces our drug therapy into our simulation. Now you'll notice at line 133, this is inside the step function for the viral replication steppable. And so when we insert code into this function, that will be executed at every simulation step. So let's start by defining our variables, our model inputs to describe our drug therapy simulation. Specifically, the time in simulation when the therapy is applied, and the magnitude of the reduction that's imposed by the therapy. So from line 133, let's just hit return a couple of times and make a little bit of space. Notice on my screen now, from 132 to 135, I've got a little bit of working space now. I'm going to input a little bit of code before this comment that says do viral model. Before the execution of do viral model, at every simulation step, I'm going to insert a little bit of code that defines the drug intervention. Firstly, let's do our model input for the magnitude of reduction. I'm going to define a coefficient that gets applied to the replicating rate when the drug therapy is applied. Let's call that variable mag, M-A-G for magnitude, underscore reduce. Mag underscore reduce. And for now, let's just set that equal to 0. So mag underscore reduce equals 0. Now our second variable will define the simulation step, not the physical time, but the simulation step. Step 0, step 1, step 2, so on and so forth. The simulation step at which the intervention is applied. Let's return, and let's define this variable, and we'll call it time underscore reduce. And let's set that equal to 50. So you'll note that these simulations are defined to run for 1,000 simulation steps. So were we to define our drug intervention by these parameters, if we're multiplying our replicating rate by a current value of 0, then that would mean that the replicating rate is disabled, and there's no more genomic replication whenever the therapy is applied. And with respect to 1,000 simulation steps that defines the entire simulation, applying this at 50 simulation steps is a very early treatment. Now, we'll use these parameters first to do a little bit of testing to make sure that we've implemented it correctly, which is always good practice when doing model specification. Lots of incremental testing as we go along. Now let's return again and make a little bit of space. That line, say, on my screen, it's around 136. Again, this is between specification of the model inputs for our therapy and the existing code starts with the comment do viral model. In between those, I'm going to define a conditional block to make some code that's only executed at the simulation step equal to time underscore reduce, our time of drug therapy intervention. Now in Python, this is easy. You can start on a new line by saying if, and then time reduce, and then two equal signs. If time reduce equals what? Well, we would like to do this implementation of the replicating rate only at this one simulation step, which means that we would like to execute this block when time reduce is equal to the current simulation step. And as it turns out, that variable is available for the step function of every steppable, and that value can be found in the variable MCS. You should find it for the viral replication steppable at line 124. This is passed from the simulator, from CompuCell3D, to our steppable, in which case we can say that our code block will only execute when our variable time reduce is equal to MCS. And to complete the condition of this conditional block, we'll then add a colon. So if time reduce equals MCS colon with two equal signs. Now from here we'll hit return. Should have some tabbed space. Meaning that this next line of code in the conditional should have space beyond the alignment of the condition at 136 on my screen. Now from here, I'm going to describe what happens only at our simulation step equal to our variable time reduce, and we're going to do a loop over all of our living cells and apply our coefficient mag reduce. Now to do a loop over a bunch of cells by their type looks something like this. We'll start a for loop for cell. For cell in, this is a function provided by CompuCell. It's a convenience function to generate an iterable of cells by a cell type that you pass to it. And the command is cell underscore list by underscore type. Now this is a function, and whatever cell types that you pass to it, it'll return an iterable of all cells in the simulation that have that type. Now in our case, we would like to do a loop over all the cells that aren't dead. And in our simulations, we have 3 cell types for epithelial cells that aren't dead, and those are uninfected, infected, and infected releasing, or virus releasing, rather. In our implementation, they have the following names. So we will pass the, we'll pass the arguments self dot, and then in all caps, uninfected. This is a reference to the uninfected cell type, not a particular uninfected cell, but the uninfected cell type itself. So self.uninfected, all caps for uninfected. Next, self dot, all caps, infected. This is our second epithelial cell type that's not dead. And then thirdly, our last epithelial cell type that's not dead. This is virus releasing, but in our implementation it has a different name. We'll do self dot and then all caps, infected secreting. So every steppable has all sorts of methods defined on them to make generating these sorts of commonly used lists. Lots of these are available in a menu in Twedit. At the top, you'll notice there's a menu called CC3D Python. And then there are a lot of options for doing particular things, manipulating the simulation by doing things like building a wall or stopping the simulation from Python, doing defining chemotaxis by individual cells, and in our case, a menu item called visit. And now in visit, you'll see there's an item called all cells of given type. And when you click that, it generates a template to do exactly what we just did manually. So there are lots of features like that to sort of accelerate development of models. Now, I'm going to complete this for loop. We should have something that reads for cell in self.cellList by type, each word separated by underscores. And that's a function. We're going to pass self.uninfected. Uninfected is all caps. Self.infected. Infected is all caps. And self.infected secreting. Infected secreting, all caps. And then we'll finish that with a colon. Now we hit return. Should see another tab automatically added. That's necessary in Python. Now, once we're in here, what we have is that at the simulation step equal to our model input time reduce, at that simulation step, we'll do a loop over all cells of the uninfected, infected, and infected secreting types. And for all of those cell types, we then want to apply our coefficient mag reduce to the replicating rate of each of those cells of those types. So a great question here about the convention of using all caps for cell types. Yes, so this is done automatically by CompuCell. If you follow me very quickly, just watch my screen. If I go to the XML, there's a plugin here called cell type, and this is where just the basic cell type definitions are defined. And you'll notice that for each of them, you can design a cell type, giving it a name of your preference, hopefully informatively. But you'll notice that they're not all caps here at the original specification of the types. But then when we go to Python, there are values assigned to be able to use those cell types as references to do type-specific definitions, but the names are always capitalized. And this is just a convention that we've adopted to just, it clears up a lot of ambiguity with jumping back and forth between uppercase and lowercase. So whatever cell types you define in the XML, you'll find that when you access an attribute in a steppable using the uppercase convention of that name, that this will return an integer. And the integer is the one that you define here. And those integers are also recognized in all built-in functions to do things like this that return cell types or lists by cell type. Now, in this loop over our cell list by type, over all of our epithelial cells, again, we're inside this loop at the moment that our imaginary drug therapy is being applied. Now we want to, I'm going to jump back over to the slides very quickly. We're going to access this coefficient in each cell in that loop, because one of these models is attached to each cell, and the simulation will integrate this system in each cell according to its current state. We're going to go through in this loop at our moment of drug therapy application, and we're going to modify that coefficient in each of those cells. Now, the way to do that, we are working with the variable cell in our for loop. And here's the way to do that. This is, I'm going to go a little deeper here. The command would read cell.sbml. This is a reference to the libRoadrunner instance that simulates all of our ODE systems that get attached to cells or just get attached to basic simulations. Cell.sbml, a reference to the viral replication model attached to this particular cell, is viral replication. With replication, first R of replication being capitalized. Cell.sbml.viral replication bracket. And inside this bracket, we will put the name of the replicating rate as defined in the ODEs in the software, which, as it turns out, is just a string replicating underscore rate. So this is now accessing Rmax, the replicating rate in the ODE models of viral replication for this particular cell. And we want to apply our variable magReduce that will define the strength, the potency of our drug therapy. We want to multiply it by its current value. We can do this by just doing asterisk equals magReduce. So this is just going to take the current value of the replicating rate for each cell and then multiply that current value by magReduce. With our current settings, magReduce is set to 0, so this is currently the same as turning off genomic replication. And let's hit save. So you should have 5 lines of code, 2 inputs, mag reduce and time reduce, a conditional based on the simulation step, executing a conditional block that only occurs when the simulation step, which is found in MCS, is equal to the variable time reduce. It's our moment of drug therapy. We'll then do a for loop over a cell list by type, over the uninfected, infected, and infected secreting cells, completed with a colon, and then this access to the replicating rate for the viral replication model in our particular cell. And multiplying that by our dosage coefficient mag reduce. And then let's do a little bit of good practice just above our model inputs. Let's put in a comment. And just put in what we're doing. Drug therapy. We do this for a couple of reasons. One, to remember what we tried to do in the first place, and secondly, so that others, when we share our code, can quickly pick up what we were trying to do as well. And you can annotate this as much as you like. I do like to annotate a lot. Just give basic names. Things like that. Okay. Now, let's make sure that we save. Should have blue discs all along the top in Twedit. And let's go to player and see what happens. Actually, I'm sorry, let's go back to Twedit very quickly. Back to Twedit very quickly. And let's go to viral infection VTM model inputs.py. Now at around line 14, should see a variable plot VRM data freak for frequency. Now let's set that to 1. And what this variable will do, it will enable plotting the viral replication model variables for our first infected cell in player. And when we do this, what we'll look for, so we go back to viral infection VTM stepables, what we'll look for is for at simulation step 50, we'll look for a sudden change in behavior in the simulation. So let's go to player. If you have a simulation running, be sure to hit stop. And once we've hit stop, let's hit step in the top left. And if you got no errors, then you should see that everything pops up, including a new screen, a new graphics window with a title that just says VRM. VRM, meaning viral replication model. Now, I'm going to jump on my screen, I'm going to jump back to the slides very quickly. You'll notice that there are the 3 or 4 stages of viral replication in our viral replication model. Those stages are represented by the variables U, R, P, and A. And then when I switch back over to player in my graphics window for VRM, I find that I also have U, R, P, and A. Now, again, for all of our simulations, we start with 1 initially infected cell. And when we enable plotting of the viral replication model data, what this is plotting at whatever frequency we define in the model inputs, what we're plotting is the viral replication model variables for that first infected cell. So let's hit run, and let's look for a sudden change at simulation step 50. Now, what I should say before we do this is that we will observe another change. And what we will observe is a change due to the transition from this infected cell going from infected to virus releasing. And just very briefly, we model 2 distinct phases of viral replication in phenotypes, where in the earlier stage there's no release of virus by a cell, and this is to model what's called the eclipse phase, in which early infection does not produce much newly released virus. And then at some point during the progression of infection and replication, the cell will change type from infected to virus releasing, in which case it'll begin secreting or releasing virus back into the extracellular space. We will see that change here, and we will especially notice it as related to the variable A. So let's look for that. We're going to simulate just past 50 simulation steps and see if we notice those 2 things. First should be a change from infected to virus releasing. When you see that there, you'll notice that in the cell field, the cell has turned red, and that's now virus releasing. I'm going to pause and refresh my screen. Now, I have my screen or my simulation paused at step 46. So this is just before our drug therapy. What I'm looking for is for the replicating state variable R. You'll notice that this is skyrocketing very quickly, and that's due to exponential amplification associated with the replicating rate. At simulation step 50, you should see that change very suddenly if we implemented everything correctly. So let's see what happens. And yes, so I've paused again at step 57 on my screen. You may have done something similar. Notice that there's a sudden drop in the replicating state variable R at simulation step 50, and this is due to our drug therapy in our test case disabling this term altogether by multiplying the coefficient Rmax by 0. And we'll notice if you simulate it just a little bit longer, all of the variables for viral replication go to 0. There's a bit of release in the extracellular virus, and that's due to that first infected cell that's now virus releasing all of its intracellular virus into the extracellular space. But what you'll also notice is that due to therapy that we've implemented, that it's not releasing much anymore. And that's due to the fact that replication, genomic replication, is not replenishing the virus inside of that cell, in which case when the virus is released into the extracellular space, it is exhausting the entire supply of virus inside of the first infected cell. Now, let's stop our simulation. Let's do a quick run on a more relevant, interesting case. And then we will take a brief pause to do some quick Q&A, take a brief intermission, and then get back to model development by defining our first steppables. So let's change our model inputs. This should be around line 134, 135 for viral infection VTM steppables.py. And let's change our magnitude coefficient called magreduce. Let's change that to 0.75. So this corresponds to a 25% reduction in the replicating rate from the baseline parameter set. And now to make things a little interesting, let's change our time of drug therapy in the variable time reduce. Again, this is a 1,000 simulation step scenario. Let's do 500 simulation steps. So halfway through simulation, you see a 25% reduction in replication. Let's hit save. Make sure we have blue disks all along the top and twit it. This is one of the highly stochastic parameter sets in our drug therapy parameter sweeps. Let's go to twit it. Let's hit stop if we have anything running. And let's hit run. Now when I switch over to the viral replication model data plots, if you still have them running, you should see them as well. You still see the same transition from infected to virus releasing in our first initial cell. Now we're past 50 simulation steps and I still see replication occurring. So it looks like my model inputs updated correctly. I still see lots of replication going on inside that first cell. Now chances are, before we get to our time of drug therapy application at 500 simulation steps, chances are this first initial cell will get killed, either by contact cytotoxicity, virally induced apoptosis. There are 4 different killing mechanisms of epithelial cells. So chances are we won't actually get to watch what's going on inside of the first infected cell when the drug therapy is applied. So at some point when that cell does die, this plot will just show all variables as 0. Now, since this is a very stochastic parameter set, at least in our simulations that we ran, chances are what you see on your screen is probably quite different from what I see on my screen, or at least potentially so. And this is due in part to the fact that drug therapy simulations like these, the outcomes are particularly sensitive to stochasticity early in simulation. So what we continue to find is that for these types of scenarios, that the early events in the simulation tend to primarily dictate what the outcomes will be for later drug therapies. So in my particular case, I have a fairly effective immune response that's eliminated a lot of virus-releasing cells. A lot of green still progressing though, and lots of virus-releasing cells popping up from infected cells. I'm at about 300 simulation steps, which means my drug therapy hasn't been applied yet. And while ours are running to our moment of drug therapy, Lexi asks a great question in the chat, which is if we wanted to take our new model parameters and put them into model inputs, to keep all of the model inputs together, you have to do anything special to do that. Meaning, could we have just made our input definitions in the input script, and would they just show up in the steppables? Or is there anything special that we have to do that? The answer is that everything is set up so that whatever names and values you prescribe for different variables in the model input script will show up directly in the steppables script. So the same model input lines, the input parameters, we could have made that exact same specification in the model input script, and it would show up just the same in the steppables script. Okay, so I've hit my moment of drug therapy application, and in my case, too little too late. For my simulation, most of my cells have already been at least infected, in which case applying a drug therapy at 500 simulation steps halfway through was really to no effect concerning saving this particular tissue patch from a viral infection. If you got something different, which we certainly observe in our simulation replicas, that's very interesting. And it's certainly a product of all of the underlying biocomplexity that's represented here, specifically, again, concerning the stochasticity in early events, and then all of the spatial aspects of the distributions of infection. Now, we can stop, or you can continue running it if you'd like, but this is the drug therapy simulation. This is the same simulation that we built to do our drug therapy investigations computationally. And you can see that it's 5 lines of code, which is pretty nice. And we do some other fancy stuff to automate doing parameter sweeps and whatnot to say send this to a computational cluster to vary these parameters and do lots of replicas to do these investigations for us, but fundamentally just defining the scenario is quite simple. Now let's disable it because we don't want more therapies being introduced as we continue to develop new steppables. So before we move on, let's set our variable magReduce at line 134. We just set that to 1, and around line 135 for time reduce, we just set that to -1. Now simulations are initialized for the first initial time or simulation step. MCS is 0, and so effectively what this does is never allows this to occur. Okay. Now let's do a very quick Q&A session. Take a very fast break. We have just under an hour left. I would like to give everyone at least a couple of minutes to stretch their legs and get a drink of water and whatnot. So if there are any quick questions that we can answer to be able to progress into writing our first steppable, please enter them into the chat. Otherwise, let's take a quick break.
Hey, James.
Yes, James.
There were two very good questions, one of which is how do you set the relationship between simulation Monte Carlo step time and real time? And the second one is how many agents can you run in a single simulation?
Yeah, great questions. So assigning time to a simulation step is arbitrary in that the CompuCell simulator, all computations are done without units. So whatever time that you would like to assign to a simulation step is simply a matter of implementing that amount of time consistently throughout the models that you specify. So we assign, for example, 20 minutes for every simulation step in our simulations of viral infection. And then we have expressions that define, say, the probability of internalization per simulation step, per cell, per extracellular virus local to that cell. And something that's important when doing that with respect to assigning time to a simulation step is that if you write an expression for a probability of something occurring over a simulation step, and then you change the amount of time that you assign to that simulation step, then your implementation needs to reflect that. We'll usually do this by defining a global variable. In these simulations, the variable is called S_2_MCS. And this variable contains whatever is specified in the inputs, whatever is specified to equate to one simulation step. And then that time discretization then has to be applied throughout your model equations, depending on how you've discretized your expressions with respect to time. And how many agents? As many as your computer can handle. Yeah, there is, CompuCell doesn't place a limit on how many cells you can define or how big you can make your lattice, but your computer will certainly notify you if it's too many for its preference. That's the short answer. I mean, I have a tower and I've done simulations with tens of thousands of cells. It certainly slows down as you start increasing the size and complexity of a simulation. A lot of it, a lot of those aspects have to do with really how much data you're using to define whatever it is that you are. So it may not so much correlate to so much how many cells you've defined, but how much information you've assigned based on each cell. Okay, are there any other questions? Do we need PyCharm for writing the stepables? No, absolutely not. But some of us certainly prefer it, me included. So you'll notice when I switch my screen, I have PyCharm open. You see it in the background. So depending on the work that you're doing, sometimes it's easier to develop in PyCharm for more actual software-intensive work, whereas Twedit most certainly has its benefits for all of the snippets, all of the supporting workflows that are built in to try to facilitate model development as quickly as possible using CC3D. So oftentimes I find myself having the same script loaded into both PyCharm and Twedit, and I will jump back and forth between the two. Okay, so I think that's all the questions for now. Let's do a 5-minute break. Let's let everyone stretch their legs, It's 4:10 Eastern time here. So let's say we'll be back at 15 past the hour, and we will start in on writing our first steppable. So let's meet back in 5 or so. All right, welcome back, everyone. Hope everyone had a nice stretch. It was a long run there. We've got 45 minutes left. Let's switch over to the slides and look at starting our first steppable. So one of the things that the framework currently doesn't include is recovery. And that is, of course, that when tissue gets damaged, hopefully something grows back. And the simulations that we were considering when we first built this didn't really operate over a time scale that was really particularly relevant for something like recovery. But we're going to add that now. So the event that we want to model is that damage in an epithelium recovers. And what that's tantamount to is dead cells being replaced. So what we would like to implement is something like a recovery time that takes about a week. So for a particular dead cell, should see something like replacement by a healthy uninfected cell after a week. Now, something to consider in our implementation is that our epithelial cells are frozen in that they don't move, in which case the model functionality to simulate mitosis that comes packaged with CompuCell can't be used without unfreezing the cells. We're not going to do that today. There's lots of functionality in CompuCell to be able to simulate growth and then mitosis of cells. In this case, what we're going to do in a very simple trick, as it were, would be to look at our 4 possible cell types for epithelial cells. Those again being uninfected, infected, viral, virus releasing, and dead. And what we're going to say for now is that recovery of a particular cell is a dead cell in simulation becoming uninfected. So in a recovery event for a particular cell, a dead cell, that dead cell will go back to being uninfected. Now, just as an overview of what we're going to do, we're going to create a new steppable, and that steppable is going to be called recovery steppable, something intuitive so that others can figure out what we did in the first place very quickly. We're going to add just one model input, similarly to what we did in our drug therapy simulation. And that model input will be the probability of recovery for each dead cell at each simulation step. So in our new steppable, we're going to create a step function. We're going to do another loop by cell type. In this case, we're going to do a loop over all dead cells. And then we're going to consider the probability of that dead cell becoming uninfected. And you can see a pseudo algorithm here. But let's just jump right into it. Let's go back to Twedit. And we'll start and work in viral infection VTM steppables.py. Now on the left, you can find the item for viral infection VTM steppables. And you can right-click on this particular file in the project pane. And you'll notice that the third item that pops up says Add Steppable. Now, of course, you can define whatever Python classes you'd like to do model specification. Any supporting Python classes according to what you'd like to do. But for adding a steppable, this is a rather straightforward way to do this. So again, right-click on viral infection VTM steppables.py and click on Add Steppable. Now, you can leave everything configured as it is by default. And let's add in the text field the name of our steppable, which again is recovery steppable. One word, recovery steppable. And let's click OK. Now, Twedit should have jumped down to the bottom of viral infection VTM steppables.py, and you should see a template that's been inserted into the simulation script with the proper class name that was specified, and then our init, start, step functions, and another function called finish, which we won't work with today. Now you'll notice along the top that there are two files that now have red disks, the first being viral infection VTM steppables.py, the other one being viral infection VTM.py. When we go to viral infection VTM.py, this is actually the simulation script that tells CompuCell about your steppables. And the reason that there's a red disk is because Twedit also automatically adds the import functions to load our new steppable into CompuCell. In which case we need to save here. Twedit doesn't save that, so you can undo something if you didn't like what happened. So let's save to get a blue disk for viral infection VTM.py. Let's go back to viral infection VTM steppables.py. And let's also save. Now the template that was generated includes some little example functions. For our recovery steppable, we won't need the start function, so we can just delete that. And in the template step function, let's delete the print and cell loop commands. So we should just have now an empty step function. Now, one trick that's particular to this simulation framework for this entire project is that along with the functions that are packaged with CompuCell, like cell list by type, we also define functions that are specific to this project. And so one thing that we need to do to modify this template steppable is we need to make one critical replacement using a specialized steppable that we've developed to support this project. Now the easiest way to do this is to scroll up to, say, around line 991 to the oxidation agent model steppable. And let's grab this class that's being inherited of viral infection VTM steppable base pi. You can just double click that and copy that. So copy this project specific class around line 991. And then let's go back down to our new steppable. For me, it's around line 1072. And now there are two instances of steppable base pi. We're going to replace those with the special steppable for this project. So paste there, and one more paste below in the init function. Paste. So now your recovery steppable should inherit from a class called viral infection VTM steppable base pi. And then that class should be initialized in your initialization function. Now, if everyone is with me, let's go to our step function and let's implement recovery. Now we're going to do something that speaks to a question that was asked about time scaling. This is a great example of what this actually looks like, to scale a model according to whatever time discretization is applied to a simulation step. Let's define our probability of recovery per simulation step. But let's do this with respect to a time that's assigned in the model inputs to a specific simulation step for a particular simulation. So let's name our variable for the probability of recovery. Let's call it recovery underscore prob. And let's assign to that a value that will update according to the time discretization of a simulation. And let's do this as follows. The variable that's defined in model inputs and is imported into this script that defines the duration of time for a simulation step is s underscore 2 underscore MCS. And again, these simulations, it's 20 minutes. And we're going to divide this. This is in units of seconds. We're going to divide this by 7 days in units of seconds. So let's divide and start a set of parentheses, and 7 days times 24 hours per day times 60 minutes times 60 seconds. And now you'll notice that if we were to say make a simulation step amount to longer time, longer physical time, then the probability of recovery over any particular simulation step is greater. And if we had very fast simulation steps, then we would be simulating less time, in which case the probability would go down per simulation step. Now, let's do a loop over all dead cells. Let's go to the menu bar, CC3D Python. And at the bottom, let's go to Visit. And then all cells of given type will be the second item. We'll drop in a little template to fill out. Let's start by removing the comments that were inserted. Now let's go to specification of the loop and what types we're looping over. In this case, the one cell type that we're looping over is the dead cells, and in simulation they are specified with the name dying. again in all caps. So the one argument should be self.dying, dying all caps. Now in our loop, let's remove the template code that was inserted. I tweet it. And what we'd like to ask over every cell in every simulation step is whether or not the cell that's dead recovers. Now we have NumPy imported into this project, and so doing a conditional statement like this is as easy as follows. If NumPy is imported as np, so if np dot random dot rand parentheses, this draws a uniformly distributed number in 0 and 1. And we'll say if that random number is less than our recovery probability. Then recovery occurs for this cell. So at every simulation step, for every dead cell, if a uniformly distributed random number in 0 and 1 is less than our recovery probability, dead cell recovers. Now, ordinarily, a cell type change in CompuCell is as easy as follows. For a particular cell that happens to have recovered, the cell type that's assigned to that cell can be accessed with the attribute named type, cell.type. And this can be written to using the same sort of convention that's been used previously to reference cell types defined for the simulation. Now, in recovery, again, we said that a dead cell sort of resurrects and becomes uninfected. In which case we say cell.type equals self dot, all caps, uninfected. So loop over all dead cells, draw a random number that's less than the recovery probability, implement recovery, and the implementation of recovery will start with changing the cell's type to uninfected. Now, for this particular simulation, with all of the different modules that are assigned to different cells, one thing that we haven't done much work on cleaning up is adding a new uninfected cell to simulation, other than initializations. And the reason is because we've not considered recovery. And so for now, what we can do, let's add a comment under our cell type change. And the comment will say, implement cell type change. And this will do all of the processes required to get an uninfected cell behaving as it's supposed to according to all of the modules that are defined. Now, fortunately, in the very first stepable, all of this is done in one block of code. So let's go to the very top. And then let's scroll down to around line 58. You should see a line that reads cell.dict and then something to the effect of VRL key equals false. Now, we want these first 4 lines all the way down to line 61, the self.load viral replication model. And all we care about are the first 2 arguments. So all 4 lines with the first 2 arguments of load viral replication model should have highlighted something like what looks like my screen. Now let's copy all that. And let's jump back down to implement recovery. Now I'm back down in the step function for our recovery steppable, and where we annotated implement cell type change, let's paste. And with what we pasted, we need to complete the parentheses on the 4th line for load viral replication model. And that's it. So now, loop over every dead cell, test for the probability of recovery, change the cell's type to uninfected, and implement it. Let's test it. Now, we're going to go down a little bit further outside of our loop. When doing this, make sure that your next line is at the same indentation level as the for loop. Otherwise, what you enter will go inside the for loop as well. And let's put a comment here that says test scenario, and then something like reduce binding. Now, what we're going to do is we're going to set up a quick little case where early in simulation, we're going to shut off internalization, and then we're going to see recovery occur for what damaged tissue is in simulation at our shut-off time. So we don't have to formalize this with a new model input, because we're gonna, this is just for debugging and testing. So let's do a conditional. Let's say if MCS, this is the simulation step, if MCS equals, that's a double equal, since this is a conditional, if MCS double equals 200, so at simulation step 200, complete the conditional with a colon and return. Should have a new level of indentation. And we're going to change the global model parameter for the association coefficient for virus and receptors. This is a parameter that was changed in the parameter sweeps, and we played with a lot in the first workshop last week. We're going to do this programmatically now instead of with the sliders, and this is very easy to do. So if MCS double equals 200, self dot, this is shared underscore steppable underscore vars, VARS. This is a global dictionary, that's a global Python dictionary that's shared among all steppables so that information can be passed between them or into and out of the simulation through a Python API. So self.shared steppable vars, let's start some brackets and a string. And the global parameter key is K underscore on. This is the value that's being changed by the sliders for your reference. So self.shared steppable vars with the key K on is equal, let's say 1E negative 12. can't set this to 0 because of the formalism of internalization. It'll end up being throwing an error for division by 0, but we can set it to a very small number. So let's set that equal to 1e-12. This effectively shuts off internalization, meaning that the virus will stop spreading. Replication will continue. Virally induced apoptosis and all immune signaling will stop, or will continue, but internalization will stop. So let's set that. Let's make sure and save. I've got a blue disk here. And let's go to player. If there's anything running, we'll hit stop. And let's hit run. Now what we should see is mostly normal progression of infection according to what we've observed time and again with the baseline parameter set. Until step 200. And at step 200, what we should see is no more spread of infection. We'll most likely see infected cells becoming virus releasing due to the viral replication model. We'll continue to see release, but infection should stop spreading. The immune system, after being recruited, immune cells after being recruited, should eliminate all infected cells and leave damage in the form of a cluster of dead cells. And before our improvements that we did today, that would be the end of simulation. The cells are just dead, and that's all. But now with our improvement, what we can then start to simulate is the actual dynamics of recovery. Of course, in this case, we're just simply drawing dead cells in a list, in which case we would expect that as infected or as uninfected cells start emerging from the dead cell area, we would expect to see them popping up randomly. Now I'm going to maximize my cell field for the epithelial sheet, and I see uninfected cells occasionally popping up and then immediately going back to being dead. And the reason for this, for them dying immediately, is because of cytotoxic killing by immune cells. The primary killing mechanism through this region right now is secretion of an oxidative agent that kills epithelial cells indiscriminately. Now, as that field decays and those levels decrease, as you see here, now starting to get uninfected cells popping up at random with occasional death. Again, due to exposure to an oxidative agent. But as the simulation progresses and that level goes down of the cytotoxic, the oxidative agent, yeah, you can see this now here on my screen. Should see this sort of fill in a somewhat random distribution, but mostly from the outer perimeter where the oxidative agent is the lowest. And yeah, that's what we see here. So of course, this is again just seeding in new uninfected cells by type change at random. And so the best sort of biological representation that you can really call this is to say that there are stem-like cells underneath our sheet, and that those cells in a somewhat random distribution underneath will somehow replace the dead cells on top of them, which are the ones that we're looking at. over time. And of course, that's more or less relevant depending on what epithelium you may be interested in modeling. In our particular case, the goal is not so much to implement a lot of biological realism, but rather to get a start on introducing the mechanism of recovery in general. Because now there is a clearly defined recovery steppable that can be modified and improved, that is specifically written to implement healing of damage due to infection or otherwise. So we can stop our simulation now, or you can continue letting it run. Now, very quickly, we have 15 minutes left. And we can very quickly improve this by saying that the recovery probability of any particular cell depends on the number of uninfected cells and its neighbors, meaning that recovery of damaged tissue must somehow be locally connected to healthy tissue. And we can do this very quickly. I'm going to somewhat blast through this a little bit. You're welcome to type along or just watch. What I'd like to do is put this together very quickly, and you can see the difference and what sorts of differences can be implemented concerning the biocomplexity with minimal amounts of code. But I'll talk through it. So in our for loop over each dying cell, what I'd like to do is count the number of uninfected cells for our given cell of interest. And I'm going to do that using this code snippet and a convenience function that's defined that loops over all of the neighbors of a particular cell. Now the actual neighbor that's returned here is another cell object, or it's the medium. And if it's the medium is just represented as the Python none, in which I'll say if neighbor, meaning my neighbor has to be a cell. And then if that neighbor's type is uninfected, then my running sum of the number of uninfected neighbors increases by 1. So if it's a cell, if the cell is uninfected, then increase by 1. Now, we'll go back down to my conditional for the probability of recovery, and now we'll make this dependent on the number of uninfected cells. So this recovery probability is now per cell, per simulation step, per uninfected neighbor. So doing this multiplication fundamentally changes what this recovery probability now means. And otherwise nothing changes. Count the number of neighbors, make the recovery probability depend also on the number of uninfected cells. So let's go, if you're just watching, I'll run this simulation. If you've been following along and adding this code in as well, you can hit stop in player and play. While that's winding up, we'll just drop this entire block of code from the step function into the chat. Again, nanoHUB oftentimes is not very friendly about pasting in, but local installations shouldn't have a problem with that, in the very least. So again, 200 simulation steps. Suddenly internalization will be shut off due to our test suite, our little test command. And then once the immune system brings in immune cells and eliminates the infection, should find a nice little area of damaged cells. I'm at 120 simulation steps. And then once the simulation generates that patch of dead tissue again, as we observed in the previous test, this time instead of seeing uninfected cells popping up at random in the group of dead cells, instead we should see all uninfected cells. I have an error. There's a typo. There's a correction at line 1083. If neighbor.type equals self.uninfected. So for those of you who grabbed that pasted code, you need to make that correction. So restart our simulation. So now in the region of damage where the dead cells have accumulated, either due to virally induced apoptosis, contact cytotoxicity, oxidative killing, bystander effect, so on and so forth. What we should find is the healthy cells repopulating the damaged area, but not at random throughout the damaged region, but instead from the interface with the remaining healthy cells. Halfway there, got 90 steps or so left before internalization gets changed. See the immune response state variable going up. Should see lots of immune cells getting recruited. Here they come. See some death near where immune cells are. There's contact cytotoxicity and the bystander effect. Now, lots of signaling. Immune response state variable increasing drastically. Should see lots of immune cells flooding in. CC3D's current rendering bug plaguing me today. Now, as immune cells start accumulating, yeah, this big patch of damage. This is also due to activation of those immune cells via cytokine signaling by the infected and virus-releasing cells. Okay, so maximized my screen for the epithelial sheet. I'm going to watch this and see if this works, and then we're going to turn it back over for questions and concluding remarks. So let's look and see if we see this. So we should see in these, yes, in these interfaces between the dead and uninfected cells, and say a neighbor like this that has more neighbors of uninfected cells than say here, should see that here, this particular dead cell has a higher probability of recovery than one here. But all the same, we should see this interface filling in with uninfected cells. Meanwhile, these few remaining infected and virus-releasing cells, in the very least, will die by virally induced apoptosis. Again, you'll notice that there's no spread of infection due to our test that we added, which effectively turned off internalization. Okay, so now saw some recovery up here. There's one. There's one. Two. Yeah. Notice no more random uninfected cells popping up in the middle of the damaged region. So this is, yeah, so this is working. And yeah, it's as simple as that. Now, I had some other demos planned, just in case we got through all of this quickly. Again, I'm going to skip through these very quickly and take us to concluding remarks. But the demos that were both done today and ones that were also planned and designed and so on and so forth, again, those are available on Google Drive in the link that we included earlier. I can drop that back into the chat again. All of the simulation files for all demos are available there, as well as these slides. So you can even go through on your own time after the workshop, and even look through the descriptions of some of the other demos that have been planned. Of course, if you need any help with getting through any of those, and so on and so forth, I'd be happy to help. My contact information will be included on the last slides. I believe it's been shared a couple of times in the chat as well, so please do reach out. On the topic of reaching out and interaction and so on and so forth, as we said in our last workshop, we're very excited and interested in doing collaborative development of this platform, both in modeling and experiment. So there's lots of computational work to be done, lots of infrastructure to build. There are lots of models to build and implement. There are lots of experiments to be done. We have all sorts of things that we're interested in. So we're very social, as it were. So again, our contact information will be included. If you have anything that you might be interested in, please reach out. We'd like to acknowledge funding, both from NIH and NSF, to be able to do this work, as well as all of the collaborators who have been involved in various aspects of this project, from say the computing to more of the immunology to experiment and so on and so forth. So I'm certainly grateful for their continued support and involvement in various aspects of this framework. We have a summer course that we do on CompuCell every year. This one is being done virtually due to COVID-19. But this quite literally starts at basic Python and works all the way through independent model development and a hackathon. So if you are interested in really diving into learning how to do CC3D based Python and XML model specification and development, this is a great way to do that. We'll be doing this with pre-recorded sessions as well as interactive hackathon blocks of time remotely. You can find more information at our website, CompuCell3D.org. You can follow our GitHub project again. Everything that we do, open source, freely available. You can watch, follow, fork, so on and so forth. and the link is in the chat. With that in mind, here's our contact information for both Dr. Glazier and myself. I'd like to open this back up to all of you for last minute questions, comments, remarks, so on and so forth. So again, please enter them into the chat. We'd love to hear from you. So James, I think I see some talk about another session.
It seems that way. If people have interest in having another short workshop of this kind, let us know and we'll see whether we can put something together that would be of interest to people. So let us know what you're interested in. The summer school in late July and August, of course, is 2 weeks, and that covers things in a lot of detail that you can't cover in 3 hours. But we're happy to work on another session of this kind as well in the meantime.
Absolutely. Okay, well, thank you, everyone. Yeah, again, please, our contact information is here. We can definitely begin discussion about perhaps doing a follow-up session on this with more demos and so on and so forth. For now, taking up a lot of everyone's time, so let's part ways here. I want to say again, speaking for everyone who's involved in putting this on, we're all very honored to have all of you here, and we're very grateful for your interest in this work. Love to hear from you soon to talk about either future work, current interests, or future workshops. So we look forward to hearing from you, and thanks again. Have a great day.